So the course is analysis and interpretation of biological data. So it's about data analytics, and we'll also cover a little bit of ML towards the end. So since the principles of data analysis are themselves quite universal, nothing to do with biology per se. So we'll talk about those general principles and apply them to some biological problems, right, as and when it is appropriate. So as you know that we are living in the era of big data, and big data applications are growing every day in almost every possible domain, right? So if you look at this slide, for example, in banking, insurance, media, education, engineering and technology, is lots of them, etc. So in biology and healthcare also there are lots of applications, right? And there's so much data that very often forget about analyzing data. Even storage of data is becoming a big challenge, right? It's like life is groaning under the burden of big data these days. So what is big data? In fact, I also used to wonder, why do they call it big data? I mean, how big is considered to be big? So apparently the definition is if the size of data is more than a few thousand GBs, so big that you cannot put it on your hard disk on a desktop or something. So then you call it big data. So it's a slightly sort of precise border, but that gives an idea of what is considered big data. So when you talk about big data in terms of and you need to quantify it, because to be able to define what is big and what is small, right? The units of data, as you know, are start with bits and bytes and kilobytes. So we in our daily usage, we know kilobytes, megabytes, gigabytes, and terabytes also are these days commonly used. You see this terabyte hard disk floating around. The next thing is each of them are simply related by a ratio of 1000, or more specifically 1024, right, to 2 power 10. So 1000 terabytes is called one petabyte, and then 1000 petabytes is a exabyte, and then 1000 exabytes is zettabyte. It's not zettabyte, it's zettabyte. Then next thing is yottabyte, then brontobyte, and geobyte, and so on. You see that the numbers get very big very quickly. To give an idea of how big these numbers are in comparison to our daily experience, so 1 exabyte is equal to 36,000 hours of a high-density video. Okay, so it's like maybe 18,000 movies or something like that. And if 11-ounce coffee cup on your desk, right, is equals 1 gigabyte, 1 zettabyte will be equal to the volume of the Great Wall of China. So that's these are the kinds of numbers we're talking about. And if you look at internet traffic, right, Cisco estimated, is an older article, estimated that by 2019, internet traffic will hit 2 zettabytes because traffic is growing at the rate of 23% annually. And in a more recent article, 2021 article, it says traffic will reach, I think this is in 2020 or something like the article. The traffic will reach annual rate of 3.3 zettabytes. So in telecom also, you see we've been seeing this kind of exponential growth in terms of data sizes. So 5 billion mobile phone subscriptions worldwide, right, and 3 billion people accessing the internet. So 281 petabytes of telecom traffic in 1986, right, growing steadily, exponentially. 67 exabytes annual. I couldn't get more recent data on the on Wiki also. Now, video data, as you know, takes up a lot of space, and video coverage has been increasing a lot. You have CCTV cameras everywhere. India also, it's picking up a lot. To give some data from UK, the 5.9 million closed-circuit TV cameras all over the country of UK, and London is considered one of the most surveillanced cities. Right? I've seen a video or a documentary some time ago that in London, if you are walking on the streets of London, as you go from street to street, you can be tracked on a series of CCTV cameras. They have algorithms for doing something like that, assuming there's not too much noise. So, so there are 70,000 sensitive locations, right? Such as schools, hospitals, and care homes. So that's very dense coverage of by video. One camera for every 11 people. Okay, so and so you have issues like privacy. Then if a camera is pointed to some apartment and then towards the window of some apartment, you need to make sure that that part of the image is kind of filtered out and all these things. So that's what happens if you have too much of video surveillance. In the area of astronomy, physics, again, there's huge amount of data generation. One interesting project is this project called Square Kilometer Array. It's a radio telescope, right, which is spread over large area. Because the idea of radio telescope is radio frequencies have very large wavelengths. So the telescope size also will be accordingly quite large. So you cannot create one big antenna like of a square kilometer size. So they have lots of smaller antennas. Create a large array of them spread over a large area over square kilometers, and that whole array of antennas acts like one antenna basically. So this one, the SK telescope, video telescope, is so sensitive that it can detect an airport radar on a planet 50 light years away. Right? That's because it's so large in area, and it generates 750 terabytes every second. Okay, so it's very easy to generate data. In fact, even neuroscience, which is my field, people boast of generating gigabytes of data every day, and we don't know where to put our hard disk and so on. So the real challenge is not generation of data, because the technology to generate data, the experimental methods to generate data, they have been advancing at a rapid pace. But the real challenge is how do you make sense out of this data? How do you analyze them? Right, that's where the real challenge is. Now, data has become so important these days. It's now considered the fourth pillar of science. Right, in science, we know traditionally there are two foundations: theory and experiment. Right, and even theory and experiment, actually, experiment first came, and theory came much later. Because if you look at, for example, the case of astronomy, if you look at history of astronomy, people have been tracking planetary motion and star positions in the in the sky for millennia, right? But fitting them using mathematics and predicting when the solar eclipses will be, lunar eclipses will be, and when the stars will be at a given time, all that came much later with the development of basic geometry, trigonometry, algebra, and all that. And if you look at further, more recently, if you come back to recent times when Newton worked out his mechanics, right? Then we could even explain the whole astronomy, all the movements of the planets, heavenly bodies, using mechanics and not just as a bunch of curve fitting tools. We could explain why they are moving like that and not just fit curves to that and predict when they will be or where they will be next. So experiment has a history of a few thousand years at least, whereas theory, especially, so theory, by theory we obviously mean mathematical theory. That's we had a weak theory for again a very long time, at least a couple of thousand years. But the strong theory with calculus and more advanced mathematics, that's a more recent phenomenon, just a few centuries old. So experiment came first, theory came later, and computation is even more recent. As you know, it's the first large computer was around 1940s, mid 40s, the ENIAC computer, which was used in World War II to crack secret codes and things like that. And then the desktop is even more recent, right? 80s, and so on and so forth. So you know that history. So computation even more recent. So the fourth pillar is data, which is happening. We're in the middle of it. So naturally, when a phenomenon is so big, so widespread and global, there's likely to be lots of jobs, and that's the reason there is, in fact, even IIT there is a whole kind of explosion of courses in data analytics, machine learning, reinforcement learning, deep learning. There's so many courses. Every department has some kind of data analytics course these days. That's because there's lots of jobs in this field. This is again old data, but the numbers are growing every year. In 2018, in US alone, there's supposed to be there's a need for about 2 lakh big data analysts, 1.5 million managers who understand big data. They may not have worked in big data, but they should know what it is and how to make use of it. So the need is really great, and it's only increasing every year. And big data jobs in India, the number of analytics jobs has doubled since last year. This is, I'm talking about 2016. 50,000 jobs available in big data in 2017. I was not able to get more recent data because there's only data on Naukri.com and things like that. I don't take it very seriously. I was looking for more formal articles. India contributes to 12% of worldwide analytics and data science job openings, so which is quite a bit, right? Making it the largest analytics hub in the world outside the US. And a lot of big companies, multinationals, are doing a lot of leading work in this. So some more data, again, certainly old data from last year's data from LinkedIn, etc., etc. So basically, there's lots of jobs. If you take biology, big data and biology, one of the most celebrated examples of big data application in biology is the Human Genome Project. Right, this started in the 90s, and the objective of this is to sequence all the bases of all the human chromosomes. So 3 million bases roughly, right? 3 billion bases, so 3 gigabases. So then identify genes within those sequences. That means there are about 25,000 to 30,000 genes in human genome, and locate the position of the genes and completely sequence them and all that. So this effort was a massive effort. It costed $6 billion, 1,000 scientists, and collaboration among 50 countries. Took 10 years to execute. And whereas now human genome can be sequenced in a few days on the next generation sequencing or NGS machines, which are available, quite extensively all over the world. So full genome data is being collected not only in normal cases but also disease conditions. So for example, if you take the cancer genome, right? So the combined cancer genome, normal genome from a single parent, the patient, constitutes 1 terabyte. Right? If you want to take a million such genomes, that will be 10 power 18, 10 power 18 bytes. If you want to look at cancer genomes on lots of patients, then these are the kinds of numbers you're talking about. So there are a lot of centers all over the world generating this kind of a genome data on a large scale. So one is European Bioinformatics Institute, UK, stores 20 petabytes of data, and it has data about genes, proteins, and small molecules. One of the largest biology data repositories in the world. And sequence data, the sequencing technology has been improving quite rapidly. Therefore, the sequence data is also growing quite rapidly and exponentially, as you can see from this graph. This again, the graph stops at 2012, and I assume that it's growing in the same way even now. Another big place, right? Whatever China does in a large scale, as we know. There's Beijing Genomics Institute. It's called the Sequence Factory. So it has 157 genome sequencing instruments working 24/7. Right, it has samples from people, plants, animals, microbes. Generates 6 terabytes of genome data every day, right? And it can produce, sequence 1 human genome per week, which, as you know, in the first human genome project took 10 years. The same thing can be done in a week, right? And so if you look at omics, there are many different kinds of omics. Genome or genomics is study of entire genome. So here there are different kinds of genomics. There is disease genomics, that is study of altered genetics, right, in certain disease conditions. So here, if you take millions of patients in that disease, for that disease, again, you can imagine how much data can be available. Or DNA profiling, for example, in forensics, right, or to determine parenting or parentage, or to find out family lineages, pedigree, and things like that. That's again a lot of data. Or comparative genomics, to figure out how different two species are, for example, how different are the genomes. Or plant genomics, bacterial genomics, viral genomics. And in the era of COVID, right, viral genomics has become very important because people constantly sequence every new strain and compare and classify them. This is Delta, this is Omicron, this is whatever, right, etc., etc. Then the next ohm or omics is after genomics, you have transcriptomics. Right, a transcriptome is basically a set of all RNA molecules in a given cell of a population of cells or an organism. So the thing is, a single gene can produce many different types of mRNA. So the transcriptome is much bigger and much more complex than the genome that encodes it. The next level is protein, because the sequence, as you know, is gene, then RNA, then protein. Now, protein, as you know, is a chain of amino acids. There are 20 amino acids. Therefore, protein is a chain of this 20 alphabets. It's a sentence that consists out of 20 symbols. Now, then if you try to estimate how many different proteins are possible, and that's a very large number, because if you take that the average size of a protein, on average, a single protein has about 200 amino acids. The number of different kinds of possible proteins will be 20 to the power of 200, which is like a really massive number. It's supposed to be greater than the number of protons in the universe. Okay, so obviously all those proteins may not exist, do not exist in reality. These are all possible combinations, and these are just possibilities. So if you want to estimate how many proteins are actually there in realized in real organisms, the estimates go like this. There are 10 to the power of 7 to 10 to the power of 8 species on Earth, and roughly there are about 10,000 to 10 power of 5 genes per species. Right, a couple of thousand to about 10,000, 20,000. That's order. Right, and then there are so therefore there are 10 to 10 power 13 unique protein sequences, about 1 trillion protein sequences. That's a kind of a rough figure. So you see that that's a large figure in itself, but it is much smaller than the number of possible sequence. The total possible sequence space. That's a 20 power 200. That's a huge number. So the actual number of proteins that we have is a very sparse set, right, in the space of the complete space of the proteins in the sequence proteins. And even there, we are only considering proteins of size 200 amino acids. They can be much larger proteins. Okay, so we are looking at this basically very large spaces, which are only sparsely occupied, right, by real proteins. And so there are a large number of proteins to be sequenced, but the known protein number of sequenced proteins is only about 1 billion. So we try to increase a little bit more. This data is about a couple of years old, but not by a whole lot. So there's still a lot of. So we have in the 1 billion range, we need to go to 1 trillion range. So there's a lot of work to be done. So since the protein numbers are so large, right, and then a single protein itself can be so complex. So people study a single protein their entire careers. I know I was doing postdoc in this neuroscience lab, and the head of the department is an expert in acetylcholine receptor. That's one protein, and maybe it has a few variations, but I mean that's pretty much one small class of proteins. He's worked on that for 20-30 years or something like that. So that's how complex a single protein is. So obviously there's so much of data that can be generated even from a single protein. So if you look at single protein study, there are the proteins have structure at different levels. There's a primary structure which refers to the amino acid sequence. Then the protein can fold upon itself and form different configurations. The secondary structure, right, consisting alpha helices and beta sheets and all that. Then the tertiary structure, the 3D structure, and then the quaternary structure where we have multiple protein subunits coming together forming a protein complex. Okay, so there are levels of complexity even inside a single protein. So there's a lot to study even in single protein. And subproteins are very popular because they're very important, involved in some important disease classes. Like for example, the P53 protein, it's called the tumor suppression gene associated protein. The protein associated with that gene, right? It suppresses tumor. So obviously you are interested, to know why how does it work and what is its action, how can you trigger its action, amplify its action, things like that. So its mutation database, there is a mutation database for this protein, and on this one protein alone, there are 60,000 publications. So this gives an idea of what kind of staggering information is, size of information is involved when you operate at molecular level in biology. So this whole study of this full complement of proteins expressed in a given cell, an organ, or an organism, it's called proteomics. This whole field is called proteomics. Next, we go to interactome. That is, until now we're talking about just all the genes, all the proteins, and things like that. Interactome is how do all these molecules interact in a given cell? The interactions, are not confined to only genes or only RNA, but genes interact with RNA, RNA interact with proteins, and there's feedback again from protein to RNA, RNA to back to genes. Right, gene expression, that is. Right, you call this a gene regulatory network. And all these again interact with small molecules in the cell, right? And there's a lot of interactions among different organelles, like Nissl bodies and Golgi bodies and endoplasmic reticulum. So the interactions of all the molecules in a given cell is itself like mind-bogglingly complex, and that is called the interactome. Okay, so now that's all molecular level stuff. Let's get to cell level. So since I'm familiar with neurons, neuroscience, I'll give examples on this, right? And if you go to cell level, lot of a typical cell, you can think of it as some kind of a fluid-filled blob, right? It has the cytoplasm, which is basically like a gel, and covered by a sheet of cell membrane, the lipid bilayer membrane, and the inside that you have nucleus, inside that you have nucleolus, and then you have endoplasmic reticulum, and if it is muscle, you'll have sarcoplasmic reticulum. And then you have Golgi bodies, Nissl bodies, and mitochondria, a bunch of mitochondria floating around. So any cell will have this standard paraphernalia, but on the whole, the appearance of the cell looks like a blob, right? A shapeless blob. Some cells have slightly interesting shapes, like for example, certain bone marrow shells have interesting projections sticking out in different directions. They look like kind of star-like, right, projections sticking out from the cell body. Or if you look at the RBC, it looks like a donut or a vada, right? So some cells do have interesting shapes. So that gives you added dimension. How do you study that? How do you encode all that? How do you quantify all that? But if you go to neuron, it takes this whole question of cell complexity to a new level, because neuron doesn't look like a shapeless blob. It has very intricate shapes. Actually, a single neuron looks like almost a shrub, like a whole tree, right? Because it has a lot of wires sticking out, right? And if you want to encode and mathematically quantify all that, there's a lot of information. So look at, for example, a neuron as a cell body proper, like any other cell, right? Which is called a soma in a neuron, and then you have wires sticking out, and these wires on one side are short and more kind of uniformly distributed in all directions. Roughly, right? These are called the dendrites, and the other side you will typically have one long fiber coming out of the cell body, and that fiber is called the axon, and that terminates at the other end into a bunch of small branches. These are called the axon terminals, right? And so the geometry, the morphology of the neuron itself is quite important to its function, right? Depending upon its shape and its branching pattern, it encodes certain kinds of functions. It performs certain kinds of information processing functions. So to encode all that, when you try to encode all that, it's a lot of information, and people are trying to do that, and there's a lot of progress in that. So, for example, there is a neuron atlas which maps out the morphologies of different kinds of neurons. So that is, they calculate and measure the branching patterns, the lengths of number of all the fibers, their lengths, their thicknesses, the branching patterns, and the pattern in which the thickness decreases as you go from the cell body to the terminals, etc. And then their conductivities and the distribution of various ion channels along these fibers. I mean, name it, and they've measured it. So it's a practically infinite amount of data just at the level of cell morphology, and you're talking about only neurons, right? So So when you come to the cell body level, whole cell level, also you have so much information that is possible. So this gives a picture of an idea of what kind of cells are available, and so this neuromorph is the neuronal atlas I was talking about. So this pie chart tells you how many different, how many cells and examples, instances are available in this atlas of different kinds of neurons. Because neuron is not a monolithic thing. There are many different subcategories. It's a large family, but many subfamilies of neurons. So different numbers of cells available in different subfamilies. Now, if you go to from cell to network level, right, the next question of interest will be in a nervous system or in a brain, how are the cells, how are the neurons connected, right? Which neuron is connected to which other neuron? So you can think of the brain or a nervous system as a graph, right, of neurons, where certain neurons are connected to certain other neurons. So if you want to characterize this connectivity pattern, right, you need to find out if your nervous system has n neurons, is the ith neuron connected to the jth neuron? Okay, so this problem is called the connectome project. Working out the entire connectivity pattern of a given nervous system or a brain is called a connectome. So human brain has 100 billion neurons. More recent estimates place this number at around 84 or 86 billion neurons. And in that, a given neuron has about 1,000, 10,000 connections, and some of these more richly wired neurons, like the Purkinje neuron, has about 1 or 2 lakh connections per neuron. So you are looking at a very large number of connections. So 100 billion, that is 10 power 11 neurons, and about 10 power 14 or 15 connections. So to work out all those connections precisely at single neuron level is obviously a mind-boggling challenge. So what people have done is started with much smaller organisms, like for example the C. elegans, what is more popularly called a nematode. Dr. K. Subramanian in our department, he works on nematode, and if you want, you can try to peep into his lab and take a look at this nematode. They're so small you have to see them through a microscope. But so these small creatures have a nervous system of size just 300 neurons, or 302, which is a very, very precise number. And these neurons are connected by about 7,000 connections. So this network is completely mapped. So the connectome for a nematode or a C. elegans is completely mapped. A lot of interesting studies can be done on this, right? But the complexity, information load quickly explodes when you go to human case. So in human connectome project, what people are doing is not try to work out the connectivity at single fiber and single neuron level, but at more kind of a gross level. So there is the instruments like what is called the diffusion tensor imaging, which tells you if a given brain region is connected to another brain region and how many nerve fibers, right? What is the thickness of that bundle of fibers connecting brain region I to brain region J? And typically the number of brain regions will be a few hundred or a maximum 1,000 brain regions, so which is reasonable, it's tractable. Okay, so there is one project like that, human connectome project. But this connectome project is not at single neuron level because that's much more in the future. It's not going to happen anytime now. So for 68 subjects, it's about 1.8 terabytes, which is not a whole lot. But people have been dreaming of working out the entire human connectome because the idea is this guy called Sebastian Seung at MIT has written a book called Connectome, where he says that if you can work out the entire human connectome and encode that, right, then maybe in the future, if we can figure out how to reconstruct a brain based on connectome information, maybe you can recreate a person who is dead, right? Those kinds of very fantastic ideas are being tossed around. So connectome project is very exciting. It's at least it has got a lot of attention. So if you look at this whole hierarchy that we have discussed, at the lowest level you have the genome and the gene-related data. Next you have transcriptome. Next you have proteome, and then you have cells, then cell networks, then tissues and organ, etc., etc. And if you're looking at brain, you have one more level of complexity, which is the behavior. That is, okay, if you have brain which has all these levels of hierarchy, how does it produce output and how does it process the input, all right? And that itself is quite complex. So, so data analytics would mean that to understand the information at all these levels and work out the relationships among the different levels, right? Which is really mind-boggling. And so obviously we are quite far from that complete story, but there is a movement in that direction. In healthcare, again, there is a huge explosion of activity, and in India also a lot of startups are in this space, and the numbers are growing quite rapidly. Right? There's an article which talks about different domains of application of big data in healthcare. Like for example, suppose you have the problem of staffing a given healthcare center, like a hospital. Right? How many nurses you want to put in a given ward? Right? If you put too many, then you will waste money. Right? You'll be losing money. Right? Because all the beds are empty and you have just nurses walking around doing nothing. You'll be wasting money. If you put too few, suddenly if you are hit by a pandemic like this and you are short of your medical staff and people are dying, and obviously it won't look good. So how do you adaptively decide your staff strength and things like that? You can use big data to do that. That's one example. So similarly, if you look at biomarker hunt, right, for different diseases, if you want to diagnose early so that you can intervene and kind of stop the disease from going to the next stage. How do you intervene? How do you diagnose at an early stage? Because external symptoms may not be good enough, right? So if you take Parkinson's disease, for example, a neurodegenerative disease, people are looking for biomarkers in biochemical kind by analyzing blood and CSF and saliva, etc., etc. Or clinical biomarkers, that is by clinical observations. For example, we have a project in mind. I was trying to get some funding, still haven't got that money. But so if you look at Parkinson's disease, people characterize the disease using something called the UPDRS score, Unified Parkinson's Disease, blah blah blah, big abbreviation. So basically, in this, the doctor looks at the patient and then makes some direct observations and notes down that the level of impairment in motor function, in language and cognition, memory, etc., etc. Right? So point is, it's very subjective because it's purely based on observations. Whereas imagine what will happen if you measure data clinically from the patient and analyze the data and get a better understanding of the level of impairment. Like for example, in Parkinson's disease alone, because of loss of cells in this tiny part of the brain called substantia nigra, there could be symptoms seen in all the 4 major domains of brain function. Like for example, sensory motor function, cognitive function, then affective function or emotional function, and autonomic function, that is function of the internal organs. So if you want to know the level of impairment in all this, then it's better to create very precise clinical tests. Like if you look at motor function alone, there could be impairment in handwriting, in speech, in eye movements, in reaching movements, in walking movements or gait, right, etc., etc. So you can measure all these things simply by even by using just a smartphone. Right, you can strap on a smartphone to your legs and make the patient walk up and down, and the accelerometer and gyroscope on smartphone will let you measure the gait activity. Or similarly, you can take handwriting from the smartphone if you have a stylus, or you can simulate decision-making functions, right, which is a cognitive function, again on a smartphone by using a simple video game. So a lot of things can be done using simple smartphone, and there's lots of variables are becoming important, and this combination is is the basis of a huge explosive field called neurotechnology, right? And so using technology to measure parameters from patients of a given disease, and using these parameters and doing data analytics on that, for any diagnosis of disease. It is a huge space, right? Lots of startups are in this space. You can look at that if you're interested in that. So another example is using virtual reality-based gaming to detect Alzheimer's, early detection, and not only for the detection or diagnosis. Can you use such gaming as a means of therapy to slow down the disease progression? So that's also another angle to this. And here also again, when the subject or the patient plays the game, you can get some numbers out of it, get some biomarkers, and then maybe determine the stage of the patient and things like that. So we are doing one such project in the area of stroke rehabilitation, where we have developed a game, a video game. And as the patients play this game, we measure lots of parameters, and we see how the parameters change. As a patient work on this, there's actually a whole package of games. Each game looking at different aspect of the patient's function. So there's one which looks at memory, one which looks at movement and response times and things like that. So as a play this, we want to see how the parameters improve, and we also combining this with what is called neuroimaging. So we take functional imaging of the brain and see how the blood flow patterns change, because stroke is a disease of impaired blood flow, right, or arrested blood flow. So as a stroke is the patient is recovering from stroke, you expect to see some level of improvement of the circulation, or at least around the damaged brain region, right? So we want to combine this neuroimaging and gaming, right, as a means of rehabilitation for stroke patients. So you can see that the domain is practically infinite, right? Both in basic biology and in medical applications, it's just explosive. So data analytics can be very useful. So now coming to the course proper, right? So I've given you the big picture and where the domain is and where you can use it and what kind of jobs are there and all that. So the course starts with the question of what is data and what is the data object? How do you represent them? Then, so when you have multiple objects, how do you compare them? And how do you determine if they are different and how different they are? Then once you have a way of saying that two objects are different and how different they are, then can you group or cluster these objects based on that concept of similarity? Then how do you assign objects to classes? So for example, if you have a bunch of data, some clinical parameters that you measured from a patient, right? Now, can you say whether the patient has disease or not, right? That is, can you put the data object into class A, which is disease, or class B, which is no disease, which is what we are doing every day these days. When you take a COVID test, that is exactly what you're doing. You're taking some data and you're classifying it into either class A or class B. And then how do you compare groups of objects, right, etc. So that is initial, so some of the initial things. Right. The course will start with some mathematical preliminaries. We start with some basics in linear algebra, right, and derivatives in higher dimensions. But we all know how to do dy by dx. And how do you do dy by dx in high dimension spaces, right? Especially if a function maps a real number onto a real number. But if it maps a vector onto a real number, how do you differentiate it? If it maps a vector onto a vector, and how do you differentiate? How do you define derivatives? So we need all these things to be able to present our algorithms, which we'll be using in data analytics. Then linear displace problem and optimization, Lagrange multipliers, probability and Bayes theorem. So these are some of the math preliminaries we'll discuss from next class. Then we'll talk about unsupervised learning algorithms or unsupervised data analysis algorithms. First one is clustering, and here there are lots of clustering algorithms we'll look at. K-means and hierarchical clustering, scale-based clustering, fuzzy clustering, graph-based clustering, self-organizing map, and so on. Then we look at dimensionality reduction. We look at PCA. We look at then classifiers. We look at One broad class is prototype-based classification, which is very simple to understand but doesn't work very well. These are two examples of that. Then discriminant-based classification, we start with linear discriminant-based classification. Then we'll go to nonlinear discriminant-based classification methods. And mu networks are a primary example of that. And here we'll talk about a couple of examples, and this will lead us to deep networks and some of those more sophisticated contemporary methods in deep learning. We'll also look at support vector machines. I'm not covering Bayes in classifier. I cover support vector machines and also, of course, deep neural networks. Textbooks for data mining part, consulting this book by Tan, Steinbach, and Kumar, Introduction to Data Mining. For the traditional neural network part, I'm looking at neural networks, a classroom approach by Satish Kumar. The analysis of biological data, I'm not really looking at it because it has lots of statistics kind of stuff, and I'm not covering much of that in this course. For deep learning and applications, I'm just looking at some papers. So as and when we look at something, I'll give the reference to that. So grading, you have quiz 1 20, quiz 2 20, assignments 20, and NSEM 40. But this time I was thinking maybe we can, once in a while, I can give an article and ask you to read the article and take an exam based on that, one or two articles, right? Take an exam based on that, a short exam. So like that, we don't have to, build up tension all the way to the NSEM, right? We can get some credit as we go along. And that will also get used to reading papers, right? Otherwise, people are used to only reading class notes, which is not good enough. Because when you get into job market tomorrow, you should know how to read literature on your own and pick up skills and so on. So let me see. I'll talk to Sandeep about that, and so that way we can kind of eat away some of the marks from the NSEM, right, and move it at least 10 marks from that. Move it to the middle of the course. Right. Grading policy, RG, right? That's a famous IT philosophy. Okay, and so finally, may the data be with you.